So the first thing to say is give my disclosures because, as you say, I do work for the Statewide Biobank, so there will be a bit of a plug later for that, obviously. We've just established a small clinical trials unit at the HMRI, so I'm the director there. I consult for a clinical trials research organisation, so privately. In terms of academic and interaction with pharma, I was mentored early on to not take the money, so I don't. So if there is money coming from ad boards or speaker fees, it goes to a third party. So whether that's the hospital or ANZUP or so forth. And if there's travel support, it's in lieu of what I'm entitled to in my employment award. I don't draw any salary for any of the clinical trials that I do. The money goes straight to institutions to pay for other people. All right, so I would argue to you that clinical trials are needed. And here's your first question. So first question, what's this? Got anything in the... Got anything in the...
Can't see anything in the chat.
No?
Yes, Peter's saying that this is a cane toad, and I would agree.
Yes, and so this is why we need clinical trials, because we don't want to do things without thinking about them and that are stupid, like introducing cane toads to Australia, which has been a particularly bad idea. All right, good. Here's your next question. Everybody knows that carrots improve your eyesight. Is this true or false?
Anything in the chat? Anybody wanting to hazard a guess on whether the carrot juice is true? Yep, Peter's saying it's true. I think it's false. That way we've got both covered. Craig.
Okay, all right. Peter's saying it's true. You're saying it's false. Good. Okay. Why is it false?
I don't know. Because the evidence. Kevin's saying it's false too.
Yeah. Does anybody know why it's false? No.
Maybe not.
Okay, so the reason it's false, it was first promoted during World War II to eat carrots and leafy green or yellow vegetables rich in vitamin A that are essential for night sight. And this was MI5's attempt to cover up the fact that the English had just invented radar. So suddenly the Germans couldn't surprise attack the UK by air because the English had invented radar. And so in order to cover that up, they generated this lie that that leafy green vegetables are important. And this is a third reason why we need clinical trials. This is not really, let's not make a question out of this. This is the confirmation bias, or an example of confirmation bias. We overvalue things that confirm our beliefs, and we undervalue what the facts say. So we are all sorry and sad individuals who want to believe in things, and the challenge for us is always to try and see if the facts will actually tell us what's going on. So we always need clinical trials. So the other challenges that we face is that the reason we need clinical trials is that most of our healthcare has little evidence. If you look at a variety of online resources there to try and find information about what's got strong evidence, what's based on lower level evidence, and what's based on virtually no evidence and just opinion, you find that most healthcare, very little healthcare, is actually level A evidence. And when there is evidence, it's really very rarely concrete. So if you look at the conclusiveness of Cochrane reviews, or you look at the number of Cochrane reviews that are stable, it's a very small percentage. So probably only about 30% of Cochrane reviews on average are relatively conclusive, and only 6% of Cochrane reviews were stable over time. So we don't know a lot. And when there is evidence, much of it is wrong. So when you actually go forward and do a randomized control trial on a particular question, 1 out of 7 times you find out you were wrong. And this is a process called medical reversal. So we don't know. What we do know isn't very high level. What we do know isn't concrete, and what we do know is often not right. So it's a particularly challenging situation for healthcare providers, but also for people who use healthcare. So clinical trials are needed, and they've gone astray. So let's look at a clinical trial in theory. It's we write a protocol, we select the team, we put it together, we recruit the patients who participate, we enter the data, the statistics, and we present and publish the report of the trial, and then the data is filed and clinical trial registration has been obtained, and everybody's happy. But the reality is that clinical trials have become so complex that they defy all logic. So this is a clinical trial that's got thousands, literally thousands of steps overall, but steps at the site of the level of the group creating the trial, the group running the trial, and at the individual clinical trial site. And this is a cooperative group trial that's been summarised here. This is not even a pharmaceutical trial, which are even more complicated. So with all of that complexity, clinical trials are very expensive. So here we've got, this is again commercial trials, but you can see clinical trials are costing $20 to $30 million on average to run per trial. So we've been taken out of the realms of simplicity into the realms of these being very expensive commercial endeavours. Another problem that they've gone astray is that clinical trials are exclusive. So when we look at clinical trials that might be undertaken, and this is some data from JAMA Oncology, that less than half of people actually have a clinical trial discussed with them, and then if they've got any medical problems, they're discriminated against further. And the number of people who actually join the clinical trial is less than 1 in 10. So we don't make it a routine part of healthcare. We only do it every so often. And this is another reflection of why clinical trials are exclusive. This is a cooperative clinical trial where you have to have all of the green things and none of the red things in order to join the clinical trial. I mean, obviously some complete idiot wrote this clinical trial protocol. Oh, that was me. So in order to try and undertake some of these clinical trials, we end up following this pathway in the sort of the commercial trial template, and I think that's a really interesting opportunity for us to change how we do things. Pharma trial innovation. So when people talk about drug development, there's said to be this long lag time and lots of drug discovery and preclinical work and then clinical trials, and so this is the justification for why drugs cost so much. But we don't necessarily see so much innovation as we see imitation. So very rarely someone develops a new drug class and then everybody jumps on the bandwagon so that we get dozens of different varieties of the same thing, all from different companies, and then we try and sort it out in marketing. Here's an excellent example of that, which is a clinical trial of 2 drugs where the people making the blue drug here tried to show that their drug wasn't any worse than the people who had the red drug, and that in some tiny little facets of clinically meaningless outcomes that there was some kind of a benefit for the drug. So by measuring the side effects on the worst day possible, they found out that their drug caused less foot soreness than the competing drug. Didn't improve the outcomes of those patients, just simply tried to find some meaningless marketing opportunity, and that would have cost, $50, $60, $70 million to run that trial. Worse still, pharma trials, commercial trials mostly don't improve outcomes, and if you actually look at some of the data sets, sometimes there's no benefit at all, or some benefits that are clinically meaningless. And that's a wild thing to say, but this is some data from a manuscript that suggests that the kinds of benefits we're seeing over time and from different drug classes are very, very, very small impacts. So we've got, perhaps 2-month increases in overall survival over time, which is not really improving. So that's over 15 years of drug development from different drug classes, anti-angiogenics, cytotoxics, hormonal therapies, targeted therapies. There is a new drug class of the immunotherapy, which is a bit better than this, but we're really seeing very, very, very small increments, so very weak impacts. This is even more discouraging because when we actually look at what those trials and how they're designed, they almost never test versus a strong comparator. So what your diagram on the left shows is that the lines going between different companies' products are the lines that cross the circle. But in most cases, the trials are run internally against existing products, where the big black loops for each company are where they're testing against something internally. So people are never prepared to put the drug up against a comparator. They're only prepared to put it up against a weakened opponent. And funnily enough, the sponsor's product almost always wins. So 96.5% of the time, some of these trials report out that the new drug is better. That's statistically unlikely. Even if your trial fails, then what happens at your meeting, you take your clinical trial that might have a primary endpoint of wanting to measure X, Y, or Z. You think, oh, I want to improve survival in a big group of people with a cancer. Even when a trial fails and there's an informally negative trial, when it's presented, not negative conclusions are made. So sometimes that's just focused on a secondary outcome or a discussion point, but sometimes that's straight out repitch that the trial is a success and is positive. So it's a really good business to be in because you can't basically fail. And throughout all that, it's all of the success to push things through. The cost of drugs isn't actually getting any better. The cost of drugs individually is soaring. Absolutely soaring, and the immunotherapies are actually pushing that even further. So I've talked about why clinical trials are needed. We don't really know what we're doing most of the time. We're doing our best, very best, but we need a lot more help. I've talked about how they've tended to have gone astray and they've tended to become very expensive marketing opportunities. But I didn't want to turn it into doom and gloom. What I really wanted to focus on is how I think that process is retrievable. And this is there's little bits and pieces that I've done, but there's lots of really great examples of this throughout the literature that I just wanted to point out. So let's break it up into how they're all the facets of how they can be retrievable, all the dimensions of that. So the first dimension is the why. Why are we doing clinical trials? What's the point? Well, the point is not to have ineligibility criteria. The point is to have eligibility criteria and to include people that are in the real world in our clinical trials. And in that respect, I think we can listen to the children. When you actually look at the number of adults that participate in cancer clinical trials, and as an oncologist, I'm obviously coloured by the cancer clinical trials, which represent 50% of the clinical trial portfolio overall in any case. But most of my examples are from that space. But when you look at children's cancer, 85% of children who have cancer will participate in a clinical trial, whereas only 5% of adults. I think there's some real opportunities to learn the way that system works and to see how that we can improve adult cancer clinical trials. The other point is to think about why we do healthcare at all. And the things that we do for healthcare are to lower costs, to improve care, and to improve health, so-called triple aim. But that's been expanded now to the quadruple aim. So in our focus on patient experience, to improve the health of populations, to improve survival, to improve outcomes, we need to be mindful of maintaining costs at least, or reducing them hopefully, but we also need to take care of the healthcare professionals that are looking after it. So there's a very easy set of, if we actually go back and think about why we're in healthcare to start with, there's an easy way of refocusing why we should be doing clinical trials. The next aspect, of course, is who or whom, more properly. So who is the question? So here's an example of how science can really advance by just focusing on who. So this is a manuscript from people who study milk genomics. So the genetics and the genomics of how mammals make milk. Some of that is in humans, and some of that is in, example, ruminants who we use for our milk supply. But this is a conference where they mapped out the interactions and relationships between people in the field, and they actually studied it and actually encouraged people to collaborate and communicate better across a group. And as you can see, over time, they were able to build bigger and bigger and bigger networks. And that was the result of that was to actually improve the science. So in both of those panels, you can see that the efficiency of each of the scientific manuscripts that was produced out of these networks was increased, so that they were more cited, so they were more useful to people, and the number of publications also increased substantially after this process. So figuring out ways to build formal consortium builds trust and enables collaboration and productivity. And clinical trials is behind the eight ball here because we've been doing this for some time in other fields of science, in astrophysics, in geoscience, and genomics, and in particle physics. People have already learned to share resources, work together, agree to be a team, and the results come from that. So in one of the examples that one of the clinical trials that I'm really lucky to be working on, which is for the COGNO group, it's a trial called MAGMA. It's going to open at up to 30 hospitals across Australia. We've got a few hospitals open now. What we're trying to do is actually develop a formal consortium charter to try and figure out how we can actually remember that we're here together as a team, and why all people in the team should actually get academic credit out of it, not just the usual situation where it's one person whose name's at the top of the grant, but everybody who's there as a team member. And I think we're still in draft with this. It's still a process, but it's something that we're trying to do. So let's now turn to the what. So what methods can we use to improve clinical trials? And of course, what I'm talking about here is super high tech. We need to spend lots of money on super high tech improvements, or we could just build face shields out of stuff that's lying around. And so one of the things that people always get focused on with this kind of thing is, well, if we need lots of new technology, that's gonna cost us a lot of money. But I don't think you could have moved earlier in the year without a university media and communications team telling everybody about how someone in their university printed face masks for the pandemic. And creating face masks from pieces of plastic was necessary, but it was also a feature of a process that's known by different names around the world. So in Hindi, it's UGARD. In the US, it's do it yourself. It's hacking or bodging in the UK. A variety of different things. I love it. It's Trick 17 in Germany and System D in France. But in New Zealand, where I was born, it's called the number 8 wire philosophy. You basically take what you've got and try and use it as a flexible approach to problem solving. Use limited resources in an innovative way. And there's, I think, a lot of opportunities for us to do that in clinical trials. So let's look at existing clinical trial design. It's bespoke, it's elaborate, it's single use, it's fragile, and there are hundreds of complex interdependencies. It's like building a plastic model. It's like building a complex plastic model and then painting it. And if you don't put the stickers on just perfectly, or you paint it wrong, or you get glue in the wrong place, then you're in real trouble. It really slows you down. So to do more clinical trials, you can either spend lots of more money. So, it's one way of solving the problem, and commercial trials can actually do that. But the other way of solving it is to do pragmatic clinical trials. And so some of the designs there focus on simplicity, trying to build things that are robust and reliable, that are durable and reusable, but still remain effective and safe. But I think you can tell that from using very simple components, you can actually put things together. So let's look at some of those components. One of them is master protocols. So master protocols are certainly coming through into fashion as the way you organise the clinical research project by saying, here's what we're going to do. So these come in different flavours. One of them is an umbrella design, so that if you have different people with different kinds of diseases from the same group that you're interested in, you can design a trial that enables more people to take part because you're actually trying to test more than one question. Alternative, you can do a basket design where you might be interested in a particular intervention that you've got, and then trying to spread that intervention across a broader population of people who might benefit from your proposed intervention. And again, I'm focused on oncology drugs here, but I'm using words to say that those interventions or those disease states could be anything. It's not just about cancer. Another one is a multi-cohort or platform. I don't really know what to call this, but we're starting to see this more, where you're actually interested in that disease group, or that group of people that you're willing to serve. You look after a group of people who tends to have a particular illness or disease, and rather than focus on one question, you build multiple questions into the same clinical trial platform. So that if someone comes along with, in this case it's lung cancer, if they come along with lung cancer that's still just in the lungs, you might ask one question. If it's spread to the lymph nodes in the middle of the chest, you might ask the second question. If it's spread around the body, you might ask a different question. And then finally, there's the multi-arm, multi-stage design, which is something that I've become particularly interested in, where you, again, you have a group of people who might be suffering from a particular illness, and you establish a standard of care control arm, which is the best thing you can do, and you start adding different experimental arms. You might start off with two or three different experimental arms, but you compare them against the one standard of control arm. So you get 2 trials for the price of 1, and you actually enable more people to take part in a experimental arm rather than just being randomized to the control arm. It also, by being multi-stage, if you get through to an idea, you get to a time point you decide that an idea is stupid, you can drop it and add and then add different questions. So you can keep the trial going rather than doing a whole lot of work, set it up, do it once and then stop. So all of these kinds of ideas increase efficiency. The next idea is to try and improve the way that we communicate to patients. And so here, this is an example where you can use better audio-visual means to communicate the outline of a trial. Now, this is a trial that I've got running at the Mater, and this is the video, this is the slides of the video that I've made to describe the trial and why we're doing it and what it might take, what we might need to do in order to take part. And you can actually pay a lot of money for these, or you can just sit quietly on your computer at night time and just build PowerPoint slides and animate them. So this doesn't need to be terribly complicated, but you can communicate a lot of I hope you can communicate a lot of valuable ideas. And certainly this trial is running really well, and people understand the philosophy of the trial, and we've been able to engage participants really quite well with it. In order to improve other processes, I'm doing a bit of plug now. We've worked really hard for OHMR to develop a consent toolkit for biospecimen research for the Statewide Biobank, but also to try and provide a resource for clinical research moving forwards, where we've tried to develop a consent toolkit that uses plain language, that doesn't shy away from discussing the complex and difficult aspects of donating specimens for bioresearch, but still tries to get that into a format that is understandable by people. And we're trying to develop audio-visual resources for this as well. And then the stretch goal is to move from English to the other languages that are spoken in our community. And to complement that, health pathology, and Amanda Kuglenberg in particular, we've been focused on trying to develop a coronavirus biobank out of routinely collected samples. But in order to do that, we recognise we need an electronic consent process. So New South Wales Health Pathology has developed the back end for this, and we hope to be able to launch this soon, and then also be able to use that as a value add for people using health pathology and the Statewide Biobank as ways of engaging people for clinical research. The next thing I wanted to talk about is data management. So when you do a clinical trial, you generally have to pay a lot of money to have a clinical trial database. But the alternative is to actually use databases that are freely available, and REDCap is a really excellent example, and I don't need to sort of tell anybody that, but it's a really useful example of where one community's decided that they need a really great idea, and then instead of keeping it to themselves, they give it to the world. So Vanderbilt built REDCap, and they give it away for free. You have to learn how to use it. It's not too complicated, but it enables research and enables you to store the data yourself. So it's one of those amazing things that has led to better pragmatic clinical trials. And there are a few future adaptations of it. For example, they've developed a portal into it called MyCap, which is a app-based patient-reported outcome measure. So you can not only collect information for the clinical trial, you can actually capture information directly from participants and load it straight into the database. So all of these items are free to use. You've got to implement them, but they're free to use and they're available. I should have talked to this before, but again, the Statewide Biobank is here to help implement biospecimen research in routine practice. We have a state-of-the-art facility, but our key strength is being embedded within the health pathology collection network. So we're able to take samples from participants from around the state and enable people from around the state to collaborate together. And we've belaboured that enough, and many of you on the call are using our service. The key thing I wanted to point out, though, is that by agreement with OHMR, with the Cheryl service, collecting samples to us under the consent toolkit enables the possibility of access to administrative data linkage. And some of that data linkage is detailed enough that you can actually form the basis for clinical trial outcome measures from that data alone. Useful things that are important to patients, things like, what was the length of time that they were able to stay alive, or what was the length of time they were able to stay out of hospital. These are the kinds of information that are collected within Cheryl, and they're developing a service called BioLink, which is specifically targeted at the strategic collections, but again, with negotiation is available to be appended to biospecimens collected under the consent toolkit in the Statewide Biobank. So there's different pieces of infrastructure around, including ones that are available to the people of New South Wales, researchers and the people of New South Wales, that will enable better and more pragmatic clinical research. So finally, let me turn to some examples of what our goals might be for some of this research. So we've talked about the why and the who and the how, but let's talk about the what. What are we doing, and why are we, what's the outcome that we're hoping to achieve? What's the goal of our research? And again, I'll just plug a few of the opportunities that I've been able to take with enormous teamwork to be able to try and answer some questions that I hope are improving things for patient outcomes. So here's one clinical trial. This is the first one I was able to develop, working with ANZUP and with the NHMRC Clinical Trials Centre at the University of Sydney, which is an idea about repurposing. So in people with kidney cancer, there's a drug called pembrolizumab, which is a cancer immunotherapy drug, and it works by trying to attack the cancer with your body's immune system, and it disables some of the ways with which the immune system can fight back against, which cancer can fight back against the immune system. We thought that there was enough data to suggest another clinical trial where we took a drug that's being used for osteoporosis called denosumab or Xgeva, and combine that with immunotherapy to see whether or not the combination of the two drugs was more effective at controlling the cancer rather than just the pembrolizumab alone. So this is an example of drug repurposing. Another example is an idea of repositioning a treatment. So 25% of people with kidney cancer have so-called non-clear cell kidney cancer, or rare variants. So most people with kidney cancer have a cancer that looks like clear cell, and that's what I've described in the pie chart there. But up to 25% of people when we have first diagnosis, maybe 15 to 20% of people when the cancer spreads, have a different kind of cancer. And these people are almost always excluded from standard clinical trials. So what we were able to do with ANZUP and the Centre for Biostatistics and Clinical Trials was develop a clinical trial in two parts to see about the effectiveness of another immunotherapy drug called nivolumab. And then if that failed patients, to then think about the combination of nivolumab and ipilimumab to see whether or not that was able to rapidly rescue anybody in the situation when nivolumab had failed. So that trial's just has finished and is reporting now. We'll report the first results of it in February of next year. But that's a really great example of repositioning to some common medication or some standard medications into a novel or rarer patient population. Let's just talk about several trials that belong to other people. But for me, these are really interesting themes. So another group of people that I serve is men with prostate cancer. And there's a couple of tablets on the market that have stupid trade names that are just hormone tablets. And they're quite useful. They help men with advanced prostate cancer, but they cost a lot of money. And they always fail. They fail men within an average of about a year. And now we're relatively mature now, but they're costing the Australian taxpayer $140 million a year. So they're not an insubstantial cost. So I want to talk about three innovations with regards to these medications. So the first one is breakfast. It looks pretty tasty. Hopefully you're all enjoying your lunch. So the first innovation is that someone was smart enough. The recommended way of taking these tablets is to take them before, at least 2 hours before you eat. So you've got men, setting out their alarm to take these tablets at 5 o'clock, and then go back to bed, because you've got to take them on an empty stomach. So someone was smart enough to say, well, what if you took 1 tablet instead of 4 tablets? And they found that was just as effective as the licensed full dose, as long as you're taking it with a low-fat breakfast. You can't eat it with bacon and eggs. And it's at least as effective, or maybe actually even more effective, than taking 4 tablets at once. So suddenly you've got the same treatment. It's more convenient for the patient, and it costs 1/4 of the price. Here's another example. So this is a clinical trial where you use enough of the drug, but not too much, and then repeat and repeat. So this is 2 separate patients taking, again, this abiraterone drug. This is one of these hormone tablets for prostate cancer. The y-axis of these graphs is a representative normalized value of the blood biomarker, so PSA, so it's a blood test for these patients. And the yellow bars there represent when the patients were taking the drug. And you can see the patient on the left has had a short burst and it controlled the cancer, and then they stopped. The cancer biologically seemed to grow again, so you use the drug again and stop. And then it comes back, so you use it again and stop. And different men had different levels of need, and so one man there's taking it at quite frequent intervals, and one is taking it at less frequent intervals. But the bottom line is that using that so-called adaptive dosing compared to contemporaneous controls, and when you compare it against the clinical trials that license these drugs, it's already doubled the length of time that the cancer is controlled, which is quite an extraordinary benefit. And finally, for this is the idea of, when we take one of these tablets, when we offer men one of these tablets called abiraterone, we always recommend it with a drug called prednisone, but someone smartly pointed out that if you give it with dexamethasone, in the brown line there, you get, again, an extra benefit of up to 10 months at no cost by simply substituting one of the co-drugs that you need to use with this. So simple clinical trials can actually be, if you're focused on patient outcomes, can actually be very effective. So the ones I'm doing, I'm cheating a little bit, but the on again, off again dosing strategy made a lot of sense. So we've got one trial, we're looking at that using the other hormonal tablet, enzalutamide, and as I said, with using a video system that's recruiting really well. And we've got support from ANZUP and the Below the Belt Peloton for that. And we've also got support for the same kind of strategy using chemotherapy, not hormonal therapy. So, and this is inspired by a patient whose experiences here. He'd had a lot of side effects from previous chemotherapy, so I offered him the option of saying, well, shall we just take some chemo in short bursts and see what happens? And he said, yep, sure. So the pink triangles there are the doses of chemotherapy that he took over time. And as you can see, again, we could use the drug again and again and again to control the cancer and keep knocking it away, but we didn't need to keep using it all in the interim. The orange box there represents the median progression-free survival of the drug. So this is it's 2.3 months is the sort of the average of what men will benefit from the treatment. But as you can see, he was able to take it for 10 times that duration. And in fact, we've recently stopped him and he's joined the NZAP study. So it's an extraordinary idea that using the existing medications in a better way can improve outcomes. And just finally, the final slide I'd like to talk about in terms of data is the MAGMA study. Again, some of the innovations that we've learned from some of these trials that we're putting together. So the first thing we're doing is making sure that we're patient focused. So we're testing ideas that appear to improve overall survival in smaller trials, using chemotherapy as early as possible in glioblastoma or for a long period of time. We're using a multi-arm, multi-stage design. So the first 2 arms are those 2 early and prolonged administration of the standard chemotherapy, but we're trying our best to figure out new experimental arms to add them on. We've developed patient information videos that are about to go to ethics as we speak. We developed that consortium charter to try and make sure that we serve the membership of the scientists and the researchers who are working on the trial. And we've also made the data very parsimonious. So what we've got there is a schedule of assessments. So clinical trial schedule of assessments tells you what you have to do as the people running the clinical trial in order to run the clinical trial. And normally this is a 2-page spreadsheet, but I have simplified it a little bit here, but those are truly the activities that you need to undertake. So we've made it very simple. And the only additional activities that one would have to do on top of normal standard care are in the orange. So we're asking for as little as possible from the sites, and then therefore the amount of money that we can pay the sites is actually more representative of the work they're doing. So respecting the sites with parsimonious data is another way of making sure that the trial is more successful. So finally, I'd just like to reflect that, it takes a village to raise someone, and everybody's part of a team. And I wanted to give credit to all of the teams that have enabled me and have been part of that have enabled many of these clinical trials. In particular, the medical oncology research team at the hospital I work at, the 2 cooperative groups I'm privileged to run some trials through, the 2 coordinating centres that are supporting me, and the small clinical trials unit that we're starting here in the Hunter. Our goal is to start giving some of this back as well. So with that, I would just like to stop and be available for questions. So thanks.
Thanks, Craig. That was really great. Fantastic overview of clinical trials. I guess the challenges that face the sector and some of the really practical solutions that you and many of your colleagues are taking. Look, we've got a couple of questions already in the chat, so people are very welcome to think of other questions, but I might start with a question from Dr. Sonu Bhaskar, good friend of the Biobank. Sonu's question is, just suggest some recommendations on mitigating risks to clinical trials during COVID-19, e.g. moving to telehealth. Anything else? Thanks, Craig.
Yeah, so look, the risks to clinical trials during COVID have been extraordinary. We've seen clinical trial activity come to a screaming halt simply because hospitals needed to, at least in Australia, needed to reposition all of their clinical workflow and in clinical space for preparing for pandemic. So it's been a massive insult to the whole industry, and there's a lot of people talking about that. But like anything else, every challenge gives you an opportunity, and so the opportunity that has come through this is to recognise what's important and what's valuable for clinical trials and how we should run them better. So my team here at Clinical Trials Unit was privileged to be a little part of the national clinical trial for COVID, which is called ASCOT. ASCOT's a clinical trial that's testing ideas in people who are sick and in hospital. And we got that trial open in 9 weeks, which is it's a record for Australia to be able to open a clinical trial in such a short period of time. But even then, the UK, the English were able to open the Solidarity trial in 9 days. So we've got a long way to go. And so the move to telehealth, I think, is really, really valuable. And so I've deliberately not talked about teletrials today because we've been talking about those a lot. So the teletrials model is to try and get trials out to different communities rather than being focused on major metropolitan centres and large universities. There's some moves that are focused on enabling more commercial trials to get out there, but I think there are other ways of using telehealth, like as we're seeing here, to enable clinical trials in much more geographically diverse centres. I've got one little experiment running. I'm not ready to show the results of it yet. We're just piloting some ideas. But I think, as with anything, any challenge and adversity gives you opportunities moving forwards.
Thank you, Craig. I have another question in the chat from Kevin Spring, another good friend of the Biobank. So Kevin's he's asking about data held by Cheryl, which I realise you may or may not be able to comment on, but he's just wondering, how up to date is that data that's available for data linkage, and is there any variability based upon the origin of the data, for example, according to local health district?
Yeah, thanks. So the data there is it's not updated in real time. It is sort of it tends to flow months and or years later, but generally it's in the order of months because it's data that's needed for billing purposes. So it's the data that actually it tracks the activity of the LHDs as they deliver healthcare service, and of course they want to get paid for that, and so that variability shouldn't be there across LHDs. It's not in-depth data. It's not the kind of data that you get from, say, Kevin, and I know you're linking up from your EMR locally, but it's still a background, relatively granular data that tells you important things like if the patient is still alive, if they've needed to be in hospital, if they've needed to attend an emergency department. The goal for us with Cheryl is to actually incorporate PBS and MBS data, which will then add a further level of richness, and I think that would be a very attractive opportunity to use the health pathology collection service and the potential for the consent portal, the biospecimen collection, and then the data linkage. And with that, all of that, you've got the potential for a clinical trial just based on a few blood tests and a bit of ongoing data.
Thanks, Craig. Look, I think I might just ask a question about, I guess, the integration between biobanking and clinical trials. Could you comment on, to what extent we're really maximising the opportunities that can occur through that relationship? Do you think there's good integration between clinical trials and biobanking or not? What could we do to improve that?
Look, I don't think there's great integration, but there are, and so there's good opportunities there. So sometimes, there are very limited biospecimen collections, pragmatically in terms of what's available and what's achievable. Clinical trials are often multi-state, so there are other services like the Statewide Biobank coming. So there's an opportunity for us to think about how we might integrate with collectors around the country to enable some of those kinds of collections. But my vision is that you can actually kick off clinical trials with biospecimen collection if you're using the linkage data. So you'll be able to actually turn the tide. Instead of making a biospecimen the last thing that you think about for a clinical trial, you could make it actually the first step and the first action. And given that many of the things that we're interested in are related to clear biology, you can actually start smaller research registries from those collections. So, for example, if you're interested in screening for a particular cancer, it may be useful not to just collect blood samples from the people who are found eventually to have the cancer, but also the people who are being screened from it. And again, those kinds of things it can be incorporated.
Thanks, Craig. We have a second question from Kevin, and that is, how do clinical trial considerations change depending upon the phase, like phase 1 to phase 3? And I guess what's the impact for participants, the investigator league and pharma?
Yeah, absolutely. So look, Kevin, phase 1 is almost always a commercial. You're never really going to see investigators going into phase 1. Because the levels of complexity, if you don't know the safety of a drug, the levels of complexity are much more intensive. Phase 2 is a bit of a challenge, but it's really phase 3 and phase 4 where investigator-led studies come into their own, because you can then say you're confident that you've got a drug or an intervention that's it's safe and it's got some effect on patients, but you're looking at the comparative effectiveness or improving ways of using an existing medication. That's when it comes to the fore. And I think these pragmatic clinical trials are ones where you are improving an existing intervention or de-escalating an existing intervention or combining interventions to see if you can improve outcomes.
Thank you, Craig. I have another question that's related to, I guess, choosing the best trial. As you know, I was previously in pediatric oncology, which is a constellation of rare diseases. And in that case, often the number of possible clinical trials that exist exceeds the number of patients in which they can be activated. So I guess I want to get your thoughts on how good are we at prioritising the very best trials where, you know, we're really achieving maximum gain from the effort that we're making, and obviously subjecting patients to the best possible kinds of treatments that might improve their lives.
Yeah, so Jenny, it's sort of the other way around in the adult space. You know, there are never enough trials for everybody involved, and then those trials never get to the right participants. So I alluded to this before, that 85% of people who, 85% of children with cancer are treated under the context of a clinical trial, and it's because of these master protocols. It's the master protocols that are essentially living documents for the standard of care best treatment of a disease. I've been the beneficiary of this when I've faced adults with cancers that are traditionally seen in children, where the pediatric oncologists that I work with, or I work next to, have been able to share with me a 200-page clinical trial protocol of the absolute standard of care for rare disease X. But that rare disease X is embedded in a bunch of other rare disease variants of the same type. So rhabdomyosarcoma, or in the case that I had was a Ewing sarcoma. And all of the variations and manifestations of Ewing sarcoma were undertaken under this one protocol, which was agreed upon and to be enabled at all of the children's cancer centres across Australia, and was part of a European or a US. So there's two great big networks. So you're joining together as teams, you're agreeing to work together, you're comprehensive, such that everybody with a particular disease that comes in is incorporated in it. So there's a clinical trial for everybody and everybody for a clinical trial. And I think those kinds of lessons are the ones that we need to learn in the adult space. It means giving up being Professor Bighead, who gets to stand in front of the podium and talk about his clinical trial. It becomes more about how do we work together as a collaboration.
Thank you. Look, we've got two other questions in the chat. So Kevin's asking about why do some clinical trials fail due to poor recruitment? I can imagine that could be a long answer, but
it's a long answer. In fact, there's Kevin, I don't know if you were there was a Cancer Institute New South Wales, awards night. I was getting all of the awards. I was sitting in the crowd drinking with everybody else, and someone else was getting all the awards. But they had a speaker out who was an operations researcher from the US, a process engineer, and he talked about time. Really, time. It's all about time. You've got to get your trial open and running quickly, or else it'll fail due to poor recruitment. So one of my trials is not doing so well, and it's one that took forever to get through all the hurdles because I didn't know how to deal with many of the hurdles, and so it's the one that's taught me the most. But I'm sad now that's the one that's struggling to complete. So again, figuring out ways to capture the zeitgeist and tick all the boxes. And what we're doing with Magma is by making sure that it's focused on patient outcomes, that it has a robust design that people can see has longevity, that respects everybody taking part and shares the credit, that respects the sites and pays them for the work that they're going to do. Those are the kinds of designs that I think are really important to make sure that trials keep going.
Thanks. Yeah, no, I think we've got a couple of other questions coming in, so we'll keep going.
Yep.
So Candice Carter from the Biobank has asked whether you could speak to the ability of investigators to secure grant funding for biospecimen collection as well as analyses of the specimens.
So Candice, we've had a couple of rounds. We've seen a couple of rounds supported by OHMR for collection of biospecimens under strategic collections. We understand that there may well be future funding rounds as well. So being able to try and tick some of these boxes, I think, will be really important. How can we be inclusive? How can we be collecting biospecimens that have immediate use and then will have valuable future use? So that's one methodology. But again, the other side of the coin is, as I said, if you were thinking about ways where we can collect biospecimens and link them to administrative data, you can actually get potentially two for the price of one, or a discounted outcome. Again, the negotiation with Cheryl is really important to see what the cost of that data might be. But those, by putting those together and collecting biospecimens that you need for the trial, but also getting the data out of it, that can be a way of reimagining the way that we run the trials, as opposed to the way we're doing them currently, which is to collect a lot of, try and ask for a lot of money to support a coordinating centre, and then the sites as they're trying to recruit the patients, and then the biospecimens almost as an afterthought. So I think we have to reconceptualise those ways. In terms of analyses of the specimens, I mean, that's it's the $1 million question. Trying to find the money to do all of the analytes that we need is very, very challenging. But again, if we've got good clinical data attached to those specimens, then it may well be that we can find collaborators who are interested in sharing that wealth, and that respects the donor's intentions for those specimens as well, that they are actually doing as much work as they possibly can.
Yeah, that's a great point. I think try to maximise the utility of biospecimens, which we're very keen to progress here at the Biobank, as you know. We have a question around inclusivity from Dan Kent. He's interested in your views on why we exclude so many patients from trials on the basis of comorbidities, when those people that are excluded actually represent a large percentage of people who become patients.
And again, Dan, thanks for the question. I mean, this is really the commercial view. I took one of the slides out, but it's a slide where I compare and contrast pragmatic trials to commercial trials, which again is that idea of the kit set model versus the Lego blocks. If we're trying to measure an extremely small difference, if our goal is to register a drug and get it reimbursed and make a profit from selling a drug, then we need a small change in outcome. You don't need a big change. You need to be able to get the smallest change you possibly can, and then use marketing and sales to try and suggest that this is a game-changing breakthrough that's going to change the lives of patients. And so you need to be highly fidelitous about how you measure the difference. And so in order to do that, you need to measure the outputs very, very carefully. You need to measure them very frequently, so you've got a lot of activity in the trial. And then you also want everybody in the trial to be absolutely uniform in terms of how they're coming into the trial. They've got to be, pristine in terms of how well they are, so that you can make sure that nothing else happens to them in the meantime, so that you can show that tiny difference that you might be interested in the clinical trial. And so, again, that's why commercial trials exclude the vast majority of patients. I mean, we've seen some where the 1 in 10 is actually quite generous. Overall in Australia, as I said, it's 4 to 5% of people with cancer will ever get on a trial. And in fact, it's only 1 in 10,000 people in the United States who will take a clinical trial as their first treatment for cancer, for example. So when your focus is on the drug and making sure that you don't miss finding some key difference between your drug and your weakened opponent, then you have to have trials that exclude anybody with a comorbidity. If you're interested in improving patient outcomes, you then test the intervention in the population of patients that you wish the outcome to impact in. So again, I didn't put it up for the MAGMA trial, but I can condense the inclusion and exclusion down to 4 or 5 points, and they're essentially the same 4 or 5 points that one would use to make a decision about offering standard treatment to those patients. So it's a very representative situation, and it's because I don't care if the trial gets a positive result or a negative result. I don't care if the trial is positive or negative, because I don't need the trial to show me that the new treatment is better. Because what I need is a result that proves whether or not we should, when a patient asks, should I start chemotherapy the moment I've got my diagnosis after surgery, I can say yes or no, and both of those outcomes are equally valuable for the benefit of the patients. So if our focus is on the patients, then we should actually include the patients that we want the results to apply to.
Thanks, Craig. I'm just seeing one. I think we can possibly squeeze in the last question. I can see another question from Sonu, who's commenting that the real-world patient is indeed someone who carries multiple comorbidities, and there's an increasing trend towards including patients with multiple comorbidities in neurological cases in particular, and so argues that it's also not fair for a subgroup of patients to be excluded. But then there is clinical trial design and specificity which merits consideration. So, yeah, great comment. Thank you, Sonny. Look, Craig, I think in the interest of time, we've certainly asked you lots of questions. Thanks very much for your time and your generosity with your expertise today. I've learned a great deal, and I think I think your approach of keeping the patient, front and centre is exactly what trials need, and also that focus upon the evidence, that it doesn't really matter whether the treatment is better or worse, but let's actually have an evidence base to enable those decisions to be made. And it would be great to see, I know within clinical trials it's a big problem, unpublished trials and all that kind of things, negative trials that never get reported. These are significant issues that are being addressed partly through registries, but I think the literature is overwhelmingly biased towards the good news, and the good news isn't good news if it's not real.
Absolutely.
Yeah.
Thanks so much, Jenny. Thanks everybody for coming.
Thank you. Thanks everybody for joining us. So our next Biobank talk will be on Monday, the 9th of November. We'll have Dr. Anusha Hederachi joining us from University of New South Wales to talk about their biobanking program there. Thanks again, Craig. Thanks everybody. Take care.
Thank you.